This question is from the chapter Molecular Basis of Inheritance from class 12th textbook. Question 167, NEET 2023. Match list 1 with list 2. So list 1 has gene A, Y, I, Z. These are the genes of lac operon. And list 2 has the gene product of these respective genes. So whenever there are match questions, you have to keep an eye on the options also. Match one and check it with the options. So, number one is gene A. In the lac operon, gene A, how do you remember acetylase and A? Remember in this way. So, A is 2. Where is A2? Option 4. So, option 3 also has A2. Now, we will have to rule out between B1 and B3. B1, is it same? No, Y is not for beta galactosidase. Y is for permease. So B is 3 and beta gal is for Z. So 1. So A2 and B3. A2, B3 is this and D is 1. So our answer is option number 4. Yes. Okay. So let me give you a little clarity on this topic. So lac operon has three structural genes Z, Y, and A. So Z codes for beta galactosidase. What is the role of beta galactosidase? See, lactose is a disaccharide. Disaccharide means it has galactose and glucose. These are linked together by glycosidic linkage. So these are two monosaccharides which are linked by glycosidic linkage. This is the reason lactose is called disaccharide. So beta galactosidase is going to break this bond and this will release galactose and glucose. And then it can be used in the glycolysis pathway or any other pathway. Y is for permease. So permease is going to increase the permeability of the cell. So this is the cell. So in the environment, lactose is present. So obviously we need this lactose to enter in the cell and permease plays an important role. And then acetylase, transacetylase is one enzyme which is going to further modify it. So these three enzymes are very important in lactose metabolism. The point is of having an operon is we don't want these enzymes to be produced every time by the cell. They should be produced only when lactose is available in the environment. Why? Because transcription and translation are expensive processes. They will consume lot of ATP and the cell never waste any energy in anything unwanted. So only when it is needed, then only the transcription and expression of these genes will happen. So to block the transcription, the promoter is there is an operator which is going to block the RNA polymerase from transcribing these genes. This is the operator. Now, how operator blocks? There is another gene. Imagine it is here, and these are connected this way. So, this is I gene. I stands for inhibitor. Inhibitor. So, the product which is produced by this inhibitor gene is repressor protein. So some students might get confused because the gene Name is I, but the product of this gene is, it starts with R. So this might create some confusion. Okay. So keep this thing in mind that the product will be repressor protein. And this repressor protein will go and block the operator. This will actually physically bind to the operator, and this will prevent the transcription and expression of these genes. So, what happens when there is lactose in the environment? This lactose, through the activity of the baseline activity, is always there. Permease is, low levels of permease is always expressed, because this is the enzyme which is going to allow lactose to enter in the cell. Once lactose is inside the cell, this is going to bind to the repressor protein. And once lactose binds to repressor protein, it can no longer bind to the operator. And now there is nothing to block operator, so RNA polymerase can continue the transcription, and it will cause expression of these enzymes, acetylase, transacetylase also. And they will bring about the metabolism of lactose. When the lactose will be consumed, again the repressor will be free and it will go and block operator. So transcription will be stopped. So this is an excellent description, an excellent example of how transcription is controlled at the cellular level. That's all about lac operon. I hope I was able to give a clear picture of this concept. All the best for your exam, and thank you very much for watching my video.